Hello and welcome to another Mr. Carter Science Special. In this video we'll be looking at the history of genetics. In particular we'll be focusing on the work of Gregor Mendle and Watson and Crick et al. And their discovery of the structure of DNA. So let's get going. Gregor Mendle was a German-speaking monk born in 1822 in the Austro-Hungarian Empire, now currently the Czech Republic. And he is most famously remembered for identifying the laws of inheritance. How did he do that? Well, in the mid-1800s, Mendel carried out breeding experiments with garden pea plants. He deliberately bred together yellow varieties of pea plants with green varieties of pea plants. And then he counted the phenotypes of the offspring very carefully. He then crossbred the offspring together and he found that the characteristics were inherited in clear and predictable patterns. He then repeated his experiments by crossbreeding wrinkled pea plants with smooth varieties of pea plants. And again, he came to the same conclusion that the characteristics were inherited in clear, predictable patterns. So let's take a look at that in more detail. Here, just to help you out, I've got a diagram showing, or an image showing you green peas in a pod, and it's the colour of those peas that we will be interested in for the rest of this PowerPoint. Now, Mendel used what he called pure breeding plants. That's our plants that would always produce the colour that you would expect of them. Now, today we call those plants homozygous dominant and homozygous recessive plants, although Mendel didn't have these words at his disposal, they hadn't been coined yet. So if we use modern terminology for this, we would be saying that the yellow peas in the parental generation were homozygous dominant and that the green peas in the parental generation were homozygous recessive. And he took these two pure breeding plants and he bred them together. In the first generation of offspring, the so-called F1 generation, he realised that all the peas that were produced were yellow. So how would we explain that in our modern genetics, where we would say that using a Punnett square, the parents were dominant, were both homozygous, so the yellow pea plant would be dominant and the green pea plants would be recessive. And when we fill in the Punnett square, big G little g, big G little g, big G little g, big G little g, then we discover that we have 4 out of 4 100% big G little g, or heterozygous, and therefore 100% yellow phenotypes, as you can see in the diagram here. What Mendel did next was quite unusual, and he took a number of those plants from this F1 generation and he bred them together to see what would happen in the so-called F2 generation. And what he found was that a quarter of the peas were green and 3/4 were yellow. So how could we explain that? Again, using modern genetics and a Punnett square. Well, we have bred two heterozygous parents together. So if we fill in the Punnett square, we get big G, big G, big G, little g, big G, little g, and little g, little g. 3 out of the 4 squares contain the dominant allele and are therefore yellow. However, 1 out of the 4 squares does not have the dominant allele, only 2 recessive alleles, and are therefore green. So we would expect out of all the offspring, and Mendel counted every single offspring and took careful note of the phenotypes that he saw, we would expect a quarter of those offspring in this second F2 generation to be green, and indeed that is what Mendel had noticed. So how can we explain these results in more detail? Well, there were no green peas in the first generation of offspring, only yellow peas. However, because the first generation of yellow peas was able to produce green peas in the second generation, they must have been carrying the green colour allele, but the yellow colour allele must have been stronger. Now, Mendel didn't know about genes. He called them a unit of inheritance, and Mendel's results suggested that there were separate units of inheritance, and some of these units were dominant over others. Mendel's work wasn't properly recognised in his lifetime because chromosomes hadn't been discovered yet, but his excellent record keeping and analysis of his results meant that others could follow his results and repeat his experiments. As I've said, Mendel kept records of everything he did and he analysed his results, and this was very unusual for a time. Eventually, in 1886, Mendel published his findings. Mendel had explained some of the basic laws of genetics using mathematical models that we still use today. And the monohybrid inheritance in peas shown here is exactly this kind of thing that you need to be able to understand and explain for your GCSE course. So let's just go through this one more time. In the parental generation, we had yellow peas, which are homozygous dominant, and green peas, which were homozygous recessive, and those were bred together to produce 100% heterozygous peas, all containing the dominant allele and therefore all yellow. When we breed these heterozygous pea plants together, we find that we get this Punnett square produced, where we have 3/4 having the dominant allele and being yellow, and 1/4 of the offspring lack the dominant allele. We have homozygous recessive, 2 little g's, 2 recessive alleles, and are therefore green. Mendel's work was not accepted in his lifetime because the chromosome hadn't been seen yet. However, 16 years after his death in 1884, in 1900, people saw chromosomes for the very first time. Mendel's experiments were repeated and the published results were credited to Mendel. The movement of the chromosomes during mitosis was then observed, and people guessed that the chromosomes might carry Mendel's unit of inheritance. It was later discovered that the chromosomes were made of DNA. Then in the 1950s, Watson and Crick built models showing the shape of DNA, building on the work of Maurice Wilkins and Rosalind Franklin. Wilkins and Franklin were looking at the DNA structure using X-ray crystallography. And this model, the model of the structure of DNA could then be used to understand how DNA can replicate, how therefore chromosomes can replicate, how they can be then separated during mitosis, and how they can act as Mendel's unit of inheritance that we today know as genes and alleles. I hope you've enjoyed this short video on the history of genetics. If you have, don't forget to like and subscribe, and let me know what you've learned in the comments below. Thank you very much for your time.